process of mitosis and meiosis. Mitosis is a type of cell division in which our cell, the mother, divides to produce two new cells, the daughter, that are genetically identical to itself. It is part of the division process in which the DNA of cell's nucleus is split into two equal sets of chromosomes. The phases of mitosis consist of four basic phases: the prophase, metaphase, anaphase, and telophase, or in short, we call PMAT. These phases occur in strict sequential order and cytokinesis. The phases of mitosis is the process of dividing the cell contents to make two new cells store in anaphase or telophase. So this is the process of mitosis. In early prophase, the mitotic spindle starts to form, the chromosomes start to condense, and the nucleus will disappear. In late prophase, the chromosomes finish condensing, the nuclear envelope breaks down, the mitotic spindle grows more, and some microtubules start to capture chromosomes. In metaphase, the second process, the chromosomes line up the metaphase plate under tension from the mitotic spindle. The two sister chromatids of each chromosome are captured by microtubules form opposite spindle poles. In the third process, the sister chromatids separate from one another and are pulled towards opposite poles of the cell. The microtubules that are not attached to chromosomes push the two poles of the spindle apart, while the kinetochore microtubules pull the chromosomes toward the poles. The fourth is the telophase. Telophase is the spindle disappears, a nuclear membrane reforms around each set of chromosomes also start condense. Last is the cytokinesis. Cytokinesis is the division of the cytoplasm to form two new cells overlaps with the final stage of mitosis. It may start in either anaphase or telophase, depending on the cell, and finishes shortly after telophase. There are two cytokinesis in animals and cytokinesis in plant cell. Let's talk about the meiosis. Meiosis means in human is a division process that takes us from a diploid cell 1 with a single set of chromosomes. In humans, the haploid cell mediate meiosis are sperm and eggs. When a sperm and egg join fertilization, the two haploid sets of chromosomes form a complete diploid set of a new genome. Phases of meiosis is the cell has more complex tasks than mitosis. It needs to separate sister chromatids, and it must also separate homologous chromosomes, the similar but non-identical chromosome pairs an organism receives from its two parents. So these are the goals accomplished in meiosis using a two-step division process. In meiosis 1, homologous pairs separate during the first round of cell division, while in meiosis 2, the sister chromatids separate during a second round. This is the meiosis 1. The chromosomes begin to pair up. Second, the homologous pairs line up in the metaphase plate for separation. The third process is the homologous are pulled apart and move apart opposite ends of the cell. And last, the chromosomes arrive at opposite poles of the cell. In meiosis II, first process the chromosomes condense and the nuclear envelope breaks down. Second process, the chromosomes line up individually along the metaphase plate, and sister chromatids separate and are pulled toward opposite poles of the cell. And last, the nuclear membranes form around each set of chromosomes and the chromosomes decondense. The cytokinesis in meiosis, it splits the chromosome sets into new cells, forming the final product of meiosis. Four haploid cell in which each chromosome has just one chromatid. That's all. Thank you.